Welcome to TLDR Neuropsychology, where we discuss various topics related to the practice of clinical neuropsychology. Today we're continuing our series related to preparation for board certification in clinical neuropsychology, focused on the basic knowledge for the written exam. Today's going to be a fun day because we're going to cover some of the foundational neuroanatomy in neuropsychology. There's a lot of depth to neuroanatomy, and we're not going to cover everything completely in full depth. The goal of this lecture is to generally prime the pumps so that you have a basic understanding of neuronal structure and function and how certain systems within the brain wire together to produce certain cognitive skills. So the topics we're going to cover are just information about neurons, neurotransmitters, and synaptic transmission methods. We're going to discuss some of the histiology, which is the cellular organization of the cortex, as well as discuss how that's organized into Broadman areas, which frequently coincides with neuroanatomical function. Then we're going to just discuss some of the general structural organization of the brain, and then move into some of the functional neuroanatomical systems, things like vision, memory, language, and the frontal executive systems. And finally, we'll discuss some of the neurochemical projection systems. To start off with, this is a graphic of two different neurons, but the main structures are the same regardless of how the neuron is organized. So you can see some of the key components labeled, things like dendrites, which is where the neurons receive information from other neurons, the nucleus, which contains all of the genetic data, the soma, or cell body, the axon, which is the path along which the information travels along each neuron, and then highlighted on the axon are myelin sheath. Not all neurons are myelinated, and the myelin sheath serves to accelerate the transmission of electrochemical information along the neuron. And the gaps between the myelinated areas are called nodes of Ranvier. And finally, at the end of the neuron, you'll have the axon terminals, or terminal boutons, which is where this neuron will interact with another neuron to propagate that communication. So this is another graphic of a neuron containing many more parts, including a much finer detailed breakdown of the nucleus and the components within it, as well as some of the microfilaments and microtubules through which neurotransmitters produced in the nucleus and soma of the neuron are transported down the axon toward the terminal bouton, where the neurotransmitters can be released and interact with other neurons. Within the presynaptic membrane of the terminal bouton, you can see there's little containers which are called vesicles. And neurotransmitters are stored in those vesicles until they need to be released into the synaptic cleft that exists between the presynaptic and postsynaptic membranes. As the action potential ends in these terminal boutons, it stimulates the binding of these vesicles to the cell walls of the presynaptic membrane, causing a release of these neurotransmitters into the gap between the pre- and postsynaptic neurons. And this release of neurotransmitters is the primary means by which communication between neurons occurs. The neurotransmitters are designed to fit into specific receptors that are on the postsynaptic membrane, which can activate changes in the functioning of the postsynaptic membrane. There's another type of connection between neurons called the gap junction, which is graphicked on the right, which doesn't have a synapse and has channels that allow ions to pass directly through from one neuron to the next. But that type of connection is less frequent within the nervous system. I'm just going to highlight some of the common neurotransmitters. So these are the molecules that are released on the presynaptic side of the neuron in the terminal bouton that connect to the postsynaptic side, or the dendritic side for the receiving neuron. So there's glutamate, GABA, acetylcholine, glycine. There's a variety of polypeptides, which are just large chains of protein. ATP can act as a neurotransmitter. Nitrous oxide acts as a neurotransmitter. There's what's called monoamines, so these include catecholamines like epinephrine or adrenaline, norepinephrine, noradrenaline, dopamine, indolamines, including things like serotonin, melatonin, and histamine. These are some of the key molecules that contribute to the functioning of our brain and drive a lot of the neural activity. So as far as neural communication goes, it's important to understand the process related to action potentials. Every neuronal cell has what's called a resting potential, which is an electrical charge on the surface of the cell when there's no outside stimulation occurring. Another type of potential are local or graded potentials, which occur on the postsynaptic membrane, typically at the dendrites or soma or cell body. And these can either hyperpolarize the cell membrane or depolarize the cell membrane. So if it's inhibitory, it can make it harder for an action potential to occur or harder for that cell to activate. And if it's excitatory, then it makes it easier for that cell to activate or easier for the information to travel along that cell. And these local or graded potentials tend to be summative, which means they add on top of each other. Factors that are important to this summation include time and how close you're getting similar information. It's kind of like how waves in the ocean build on top of each other. So if you have one wave going up and one wave going down, then once they cross each other, then it shrinks the wave. But if you have both going up or both going down, then it either makes the wave much taller or makes the wave go down much lower. The action potential is the communication of information that travels down the axon, and it's electrochemical in nature. And it operates on an all or none principle. So unlike local or graded potential, which sum to determine the change in polarization, an action potential, once you hit that threshold, then you go through the whole process from beginning to end. And it's conducted through the opening and closing of different ion channels related to potassium, chlorine, sodium, and calcium. And I'm not going to get into the specific details about the ionic exchanges that occur to propagate action potentials. I think it's sufficient at this time to know that what happens is we have a resting potential, local or graded potentials build up until we reach a certain threshold. Once that threshold is met, then you get all of these ion channels opening along the axon, which causes rapid depolarization, and it's followed by a repolarization that goes well below the resting potential, and then there's pumps to exchange these ions back into and out of the cell to restore the neuron back to a resting state. And you can see the different steps of that process in the graphic to the right. Another thing worth noting about action potentials is that the threshold for the action potential can differ from neuron to neuron, and every action potential fired by a specific neuron will have the same threshold for firing and display the same pattern of depolarization and repolarization. The difference between the action potential's peak and the resting potential is the same for every action potential fired by a specific neuron. That said, the characteristics of action potentials can differ by neuron. And the communicative dimensions of action potentials include things like the rate in which the action potential is firing, the discharge pattern of the action potential. So some action potentials fire in what's called a tonic presentation, which means there's a slow increase in the rate of fire when there's a steady stimulus present. Some action potentials fire in what's called a phasic pattern, where there's a decrease or discontinuation of the action potential when there's a steady stimulus. And some action potentials fire in rapid bursts, and others have a pattern of what's called quick shot. So these patterns can change the way that a neuron is communicating within your brain. Additionally, Another characteristic that can change the functioning of neuronal communication is the amount of neurotransmitters that are released following an action potential. Some neurons can release greater amounts of a specific neurotransmitter, some release lesser. And then the specific neurotransmitter can also play an important role in this neuronal communication. Now we're going to talk briefly about histology, which is the cellular layout of the neurons, as well as Broadman's areas. So there's a variety of different types of neurons, so they're not all shaped or structured the same. So there's multiple ways to classify neurons. I talked briefly earlier about how shape can be used to classify different neurons. There's hundreds of different shapes that can be used to classify neurons. We can also classify them based off of their function, whether they're motor neurons or neurons that drive our muscular systems, or sensory neurons, which are neurons that are used to carry sensory information to and from the brain, or interneurons, which are neurons, these typically you'll find them in the spinal cord, but they connect between neurons to facilitate information travel, usually from sensory systems up to the brain. Additionally, within the brain, there are cells called glia. One type is astrocytes, which serve to generally give structural support within the brain, as well as astrocytes have been implicated in not just nourishing neurons, but also to some degree modulate the functioning of neurons. Oligodendrocytes or Schwann cells are the cells that form the myelinated sheath along the axons of neurons, with oligodendrocytes being limited to the central nervous system and Schwann cells occurring in the peripheral nervous system. So they serve the same function, but they're slightly different types of cells. And then microglia are the smallest type of glia, and they tend to be involved in facilitating immunal defense within the central nervous system and cleaning up damaged areas within the brain. And there's also ependymal cells, which form a lining between the ventricular system and the brain and spinal cord. And these are involved in the production of cerebrospinal fluid. So now we're going to talk about the histological layers that are found throughout the cortex. And a way to imagine it is it's like taking a core sample from the ground and then looking at the different layers that you'll see. And you'll find within the nervous system, different parts of the brain have a different thickness of the different layers, and that can tell you much more information about what that part of the brain is used for or how it functions. So the most superficial aspect of our brain is called neocortex, and it is in 6 layers, and you can see those layers in the graphic to the right. So the first layer up top is the molecular layer. That's followed by the external granular layer, or also called small pyramidal layer, because it does contain some pyramidal cells, but their bodies tend to be smaller than some of the other layers. The third layer is the external pyramidal layer, or medium pyramidal layer. So the bodies of those pyramidal cells tends to be a little bit larger. The fourth layer is the internal granular layer. The fifth tends to be the internal pyramidal layer, which has the largest pyramidal cells, which are multipolar cells. And the sixth is the multiform or polymorphic layer, and it tends to have a broad mix of different types of neuron cell types. There's also parts of the brain which are made up of what's called allocortex. These tend to be evolutionarily older parts of the brain, unlike the neocortex, which tends to be some of the evolutionarily newest parts of the brain. So the allocortex is 3 to 5 layers and can be divided into paleocortex, archicortex, and periallocortex, depending on the specific layout of the different cell bodies and layers. So back in the 1900s, Broadman looked at these different cell layers within the brain and noticed that there were differences for different parts of the brain. So he organized the brain based off of the cellular layout, and these areas are called Broadman areas. And these areas often overlap with certain specific sensory functioning, processing areas, as well as behavioral control areas. And you'll see in the graphics, some of those areas are highlighted, like the primary motor cortex, which controls all of your volitional movement, primary somatosensory cortex, which is associated with managing the sensations that you experience throughout your body. There's the primary visual area, which is where all visual information first travels to before being passed through other parts of the brain, as well as some other specific areas that are associated with deficits. Things like Broca's area, where damage to that can cause aphasia or difficulty producing speech, and Wernicke's area, which once again, damage to it can cause aphasia, but in their case, it's difficulty producing meaningful speech or comprehending other people's speech. In addition to the primary sensory areas that Broadman's histological map helps us identify, such as primary visual area or primary auditory area, this cytoarchitectural map also helps us identify what are called association cortex. Association cortex receives information from these primary cortical areas and facilitates additional processing specific to other characteristics related to that sensory or motor area, and sometimes involving the combination of multiple sensory areas. The spinal cord has also been divided into a histological map, and you can see some of the major tracks or pathways that the axons of the neurons travel through within the spinal cord. And the spinal cord tends to have a variety of interneurons as well. But I'm not going to go into too much depth about the spinal cord, just know that it is part of the central nervous system along with the brain. So this graphic shows some of the sensory and motor changes that can be experienced depending on where a lesion occurs within the spinal cord. A lesion being an area of damage or injury. So now we're going to get into kind of the broader general organization of the brain. So as I mentioned before, the brain and spinal cord are both parts of the central nervous system, and then we also have what's called a peripheral nervous system. So the peripheral nervous system is all of the connections outside of the brain or spinal cord. That includes things like cranial nerves and what's called ganglia, which are clusters of cell bodies within the peripheral nervous system, as well as spinal nerves and dorsal root ganglia. And the peripheral nervous system is divided into sympathetic and parasympathetic nerves and ganglia, and that just helps prime our body for either activity or to bring us back to a more rested state. And there's also the enteric nervous system, which is the highly innervated nervous system related to our gut and bowel system. And a lot of neurotransmitters are produced in the enteric nervous system and travel up to our central nervous system. Here's some of the gross structures for brain organization. So you have the forebrain, also called the prosencephalon, which is the most superficial aspect, as you can see in the graphic. And that can be divided into both the telencephalon, which is things like the cerebral cortex and the hemispheres, the basal ganglia, the basal forebrain, as well as the diencephalon, including things like the thalamus, the hypothalamus, the epithalamus. There's also the midbrain, which is below the diencephalon, and that includes the mesencephalon, as well as the cerebral peduncles, the midbrain tectum, and the midbrain tegmentum, and the hindbrain or rhombencephalon, which includes the metencephalon. Notable structures include the pons and the cerebellum, and the myelencephalon, with notable structures including the medulla. So one thing that's helped me remember some of this organization, for the encephalons that start with the letter M, they go in reverse alphabetical order when you're going from lowest to highest structure. Myelencephalon, metencephalon, and then mesencephalon, which are all reverse alphabetical. And then on top of that, you have the diencephalon and the telencephalon. Another study strategy would be to make a fist with your right hand and then wrap your left hand around the top of that fist. So your left hand would form the telencephalon, the fist would be the diencephalon, the midbrain is the narrowing part of your wrist, with the mesencephalon and myelencephalon being the bump of the muscles of your forearm. Here's another graphic that highlights some of the other important parts within the brain. So once again, you can see in blue the superficial telencephalon, and then below that you can see where the thalamus is highlighted and the hypothalamus, which form that diencephalon. And things like the superior and inferior colliculi form that mesencephalon. And then you can see where the pons are located, as well as the cerebellum, and then the medulla as it extends into the spinal cord. Another important structure is this corpus callosum, which is how both hemispheres are connected to each other. So there's white matter axons that travel between the hemispheres through the corpus callosum to coordinate the functioning of the right and left hemispheres of the brain. So as mentioned before, there's two hemispheres in the brain, left and right, and the cortical areas primarily consist of the bodies or soma of the neuronal cells. And this gives it a gray color, and it's called gray matter for that reason. And when you have clusters of neurons that have similar connections and functions, those are called nuclei. And the brain's surface has bumps and depressions or peaks and valleys, which are called gyri for the bumps or peaks, and sulci for the valleys. And those provide some helpful landmarks for identifying the different regions and anatomy of the brain, as well as serve to increase the overall surface area of cells within the brain. Axons from the cortex, gray matter, which are typically myelinated, form white matter. And these run subcortically and often project to other brain areas. And when you have bundles of axons that travel to the same area within the central nervous system, those form tracts or pathways. And if it's outside of the central nervous system, then they're called nerves. And tracts also have other names, including commissure, desiccation, or fasciculus. And some of those names depend specifically on how that tract travels within the brain. And as mentioned earlier, both hemispheres are connected by white matter tracts that form the corpus callosum. So this graphic gives a picture of some of the prominent gyri and sulci that help form the broad separations that we make within the brain. So some of the key sulci include the lateral sulcus or sylvian fissure, which separates the frontal lobe from the temporal lobe, and it goes back far enough to help separate the parietal lobe from the temporal lobe, and the central sulcus, which is used to differentiate the frontal lobe from the parietal lobe. Generally, we separate the brain into four primary lobes. So the frontal lobe, the temporal lobe, the parietal lobe, and the occipital lobe. Below the cortex, or what's called subcortically, there's a variety of prominent structures. So if you look at the graphic to the right, you'll see some of them listed that are related to what's called the limbic system. So that includes the thalamus, the basal ganglia, the amygdala, and the hippocampus. And those play a prominent role in a variety of cognitive functions, from emotional processing to memory to coordinating our movements and behavior. So when discussing neuroanatomy, it's important to understand the general orientation when describing different structures within the brain. So there's multiple terms to describe the relative locations of different aspects of the nervous system. This differs in humans compared to other animals because our brain is rotated 90 degrees from other animals, like let's say a rat. And that rotation occurs around the midbrain and diencephalic junction. So some of the key terms are things like rostral, which means head, caudal, which means tail, dorsal, which means up, and ventral, which means down. So if you look at the graphic to the right, you can see where those are in the human brain and how those rotate when we're talking about the orientation of things in the spinal cord. Additional terms include anterior, which indicates the front of the brain, superior, which indicates the top of the brain, posterior, which indicates the heading towards the back part of the brain, and inferior, which indicates the lower parts of the brain. And once again, these terms are relative, so it can vary based off of the structures that you're talking about. For example, the frontal lobes are superior to the temporal lobe, but they're anterior to the parietal lobe. Another set of terms that are necessary to understand brain organization is lateral and medial. So if you look at the graphic on the right, you'll see that medial tends to be things that are closer to the center line, and lateral is related to things further from the center of the brain. And medial and mesial are frequently used synonymously. Additionally, when it comes to information, we can talk about the direction that the information is traveling. So if information is traveling toward the brain, that information is called afferent or afferent. And if information is being carried away from the brain, that information is called efferent. So sensory information tends to travel up towards the brain in an afferent manner, and motor movement information tends to travel away from the brain in an efferent manner. And this graphic highlights how some of those terms are used to describe different gyri and sulci within the brain. So you can see there's an inferior frontal gyrus, a middle frontal gyrus, and a superior frontal gyrus, or a superior occipital gyrus, a middle occipital gyrus, and an inferior occipital gyrus. So another important concept is the sectional planes within the brain. There's three primary orientations from which we can take sections of the brain. The first is the frontal or coronal plane, and if you look on the graphic, that tends to, if you think of where a crown or tiara would sit on your head, that would be a cut vertically down from the end of the crown. The second would be the sagittal plane, which would be like separating the hemispheres in half. And that can be mid-sagittal, which would be right down the middle, or parasagittal, which would be an off-center cut, depending on what brain structures you were interested in looking at. There's also a transverse axial or horizontal plane, which would be cutting through the brain in a horizontal fashion. So now we're going to move on to some of the functional neuroanatomical systems within the brain. The networks and connections that produce or process specific cognitive abilities or process specific sensory information. So the first system we're going to talk about is related to the visual system. There's a primary pathway called the geniculostriate pathway, and if you look at the graphic on the right, it shows this pathway. And this contributes to visual discrimination and form perception. So it travels from the retinal ganglia or optic nerves, and it travels to the lateral geniculate nucleus of the thalamus, and then moves on to the primary visual cortex, or Broadman area 17. There's an alternative tectopulvinar pathway, and that tends to be an evolutionarily older pathway. So in some animals, it's a much stronger pathway than it is in humans. And it contributes primarily to pupillary light reflex, orientation to visual stimuli, and attention-directed eye movements and motion detection. So that pathway differs in that it travels from the retinal ganglia or optic nerves to the pretectal areas of the superior colliculus, to the pulvinar nucleus of the thalamus, and then on to the frontal and parietal association areas, things like the frontal eye fields. And there's an interesting phenomenon that's associated with damage to the geniculostriate pathway where the tectopulvinar pathway is intact, and it's called blind sight, where people can react to moving objects without the ability to visually see or discriminate those objects. So along the geniculostriate pathway, after things get to the primary visual cortex, which is in the occipital lobe, there's actually anatomically separate pathways to communicate the form, motion, or color of various objects. And one is called the dorsal stream, and the other is the ventral stream. This dorsal pathway is called an occipital-parietal projection, meaning that it starts in the occipital lobe and projects toward the parietal lobes, and it's preferential for processing spatial-related information or locational information. It's involved in the visual motor integration with the environment, your ability to engage with the world around you. The ventral pathway is an occipitotemporal projection, meaning that it starts in the occipital lobe and it projects towards the temporal lobe. And this preferentially processes structural and feature-based information of what you're visually observing. And that's involved in the recognition of faces and objects. Damage along the dorsal pathway can cause impairments in things like spatial perception, attention, visual motor processing, so like reaching out for a door handle and missing it, or can cause hemispatial neglect, which is difficulty processing information coming from a specific hemifield of your vision. Hemifield being a way of describing if you were to lose sight from half of what you could see, either temporally or nasally. Damage along the ventral pathway can cause impairments in things like facial and or object recognition, which is called agnosia. And there's different types of agnosia, such as apperceptive agnosia, which is impairment in processing basic visual elements of an object. And that's typically due to significant damage to the visual association areas. Or associative agnosia, which is impairment in recognition or assigning meaning to stimuli that are accurately perceived. And associative agnosia is caused by either disconnecting lesions that disconnect the association cortex from the memory systems, or can cause by less extensive visual association cortical damage. The next neuroanatomical system we're going to discuss is the memory system. So there's a few notable neuroanatomical regions associated with the memory system. So there's the hippocampus and parahippocampal areas, the amygdala, the thalamus, and the basal forebrain. And if you look to the graphic on the right, you'll see that there's two circuits associated with our memory systems. There's the medial limbic circuit, which travels from the pararhinal parahippocampal cortex onto the hippocampus, to the mammillary bodies via the fornix, from the mammillary bodies to the anterior thalamus via the mammillothalamic tract, back to the cingulate gyrus, and then back to the hippocampus. Or the lateral limbic circuit, which once again starts at the pararhinal or parahippocampal cortex, which has two projections, one to the amygdala and one to the dorsal medial thalamus. The projection to the amygdala travels along the uncinate fasciculus, and then from the amygdala, it travels up to the dorsal medial thalamus via the ventral amygdalofugal pathway, back to the orbital frontal cortex, and then back to the amygdala. The hippocampus itself is considered archicortex, which means that it's 3 to 5 layers as opposed to neocortex, which is 6 layers. And it contains the dentate gyrus, Amon's horn, including areas 1 through 4, and the subiculum. And if you look at the graphic on the right, you can see the layout of these different areas. The primary internal connections of the hippocampus form what's called the trisynaptic circuit. So it goes from the entorhinal and pararhinal cortices to the dentate, and then on to Amon's horn area 3, then Amon's horns area 1, and then back to the subiculum, and then back to the entorhinal and pararhinal cortices. Information processed in the hippocampus travels out to parahippocampal regions, which are regions that are around the hippocampus. So these include the rhinal, both the entorhinal and pararhinal cortices, the pre- and parasubicular cortices, and the parahippocampal cortex. So there's a prominent hypothesis called the two streams hypothesis, wherein the pararhinal cortex receives more anterior temporal input, so from the anterior temporal circuit, and this is non-spatially related information, and the parahippocampal cortex receives more posterior medial temporal input. So this contains more spatial related information. And the information is bound together in the hippocampus to form a singular memory episode. That said, there's evidence that both cortices receive both spatial and non-spatial information before that information is transmitted to the hippocampus. The entorhinal cortex plays an important role in that it receives mostly indirect input from the cortex, and the anterior lateral aspect receives a lot of the pararhinal and non-spatial information. And the posterior medial entorhinal cortex contains a specific type of cell called grid cells, which encode spatial location in the environment. So within those grid cells, every aspect of the environment is mapped out in a 3-dimensional map. And these grid cells project to specific cells within the hippocampus called place cells, which is how that spatial information is retained and then later encoded into our memories. Another important structure in memory is the amygdala, which is made up of paleocortex, which once again is one of those types of cortices that is 3 to 5 layers. And within the amygdala, you have the basolateral nuclei, which connect with the limbic system, association cortex, and dorsomedial thalamic nuclei, as well as corticomedial segment of the amygdala, which connects with the basal forebrain, hypothalamus, and brainstem. The amygdala has a lot of strong autonomic connections, and it's part of the lateral limbic circuit for memory. The thalamus is another important structure in our memory system, particularly the anterior thalamus, which receives information from the hippocampus through the mammillary bodies, and the dorsal medial thalamus, which receives input from the amygdala. There's also the internal medullary lamina, if you look at the graphic, fit between different aspects of the thalamus. And these internal medullary lamina receive memory-relevant connections from both the mammillothalamic and ventral amygdalofugal tracts. Historically, injury to the dorsomedial thalamic area is associated with amnesia, which is disruption of your memory functioning. That said, there's recent evidence that there's a stronger relationship between amnesia and damage to the internal medullary lamina and the mammillothalamic tract. Additionally, midline thalamic nuclei and frontal lobe connections can also play an important role in memory functioning overall. Another important area associated with memory functioning is the basal forebrain, and it has a variety of nuclei that contribute to memory functioning, including the septal area, the diagonal band of Broca, nucleus accumbens septi, olfactory tubercle, substantia innominata, found in the nucleus bacillus of Maynard, bed nuclei of the stria terminalis, and the preoptic area. And these different nuclei connect to both the medial and lateral limbic circuits, which are responsible for memory functioning. And within the basal forebrain, you'll find a lot of what are called cholinergic neurons, or neurons that release acetylcholine, which is associated with amnesia when those neurons are damaged. And there's other neurotransmitter pathways like dopamine, which may also contribute to memory function. Damage to these memory systems, as mentioned before, can cause amnesias. And there's been an increased interest in the neuroanatomy of memory and amnesia after the report of HM in 1957. So HM was a patient who had severe epilepsy, and he had a significant portion of his temporal lobes removed on both sides of his brain in both hemispheres, which included the hippocampus and the amygdala. And what they found was that HM had difficulty with what's called anterograde amnesia, or his ability to form new memories. Other surgeries done by Scoville and Milner, who had conducted the surgery on HM, found a similar pattern in cases where the hippocampus was injured. There's a two-system theory of amnesia where both lateral and medial limbic circuits must be damaged for the amnesia to occur. So what they've found is that if both pathways are damaged, people will present with much more severe amnesia symptoms than if only one of the pathways is damaged. They also found that structures within each memory pathway are highly interdependent, but that there's some redundancy between both circuits. So damage to one can be made up for by having the other circuit functioning. They've also found that extensive parahippocampal as well as pararhinal damage can produce amnesia comparable in severity to having direct damage to both the medial and lateral limbic circuits, even when the hippocampus and amygdala are themselves spared from injury. And that makes sense if you look at the graphic, where if you take out both the pararhinal and parahippocampal cortices, those drive a lot of the projections to both the hippocampus and amygdala, and it disrupts both the medial and lateral limbic circuits. Some of the common amnesia subtypes include retrograde amnesia, which is losing previously formed memories, anterograde amnesia, which is the inability to form new memories like HM, post-traumatic amnesia, which is amnesia following a head injury, which is usually considered to be temporally graded, which means that information closer to the time of the head injury is harder to remember than information further away from the time of the head injury. Transient global amnesia, which is a temporary amnesia that usually resolves within 24 hours and there's no clear cause. Infantile amnesia, which is the general inability to recall early childhood memories. That's normal for adults. to experience. Dissociative amnesia, which is memory disruption secondary to a severe psychological stressor rather than physical damage to your brain. And drug-induced amnesia, which is temporary memory loss as a consequence of certain drugs. Things like Ambien or DATR drugs can cause this. So the next system that we're going to talk about is the language system. And language tends to be what's called lateralized, which means one hemisphere preferentially processes language-related information. So 95% of right-handed people are what's called left language dominant, which means typically the left side of their brain will contain the majority of their language processing centers, whereas 60 to 70% of left-handed individuals are left-handed language dominant. The language dominant hemisphere specializes in comprehension of language as well as producing fluent speech, whereas the non-dominant hemisphere tends to be specialized in what's called prosodic information and emotional aspects of information. Prosody tends to involve things like rhythm, intonation, and how syllables within a word are stressed as we speak. And both hemispheres coordinate our speech via connections through the corpus callosum. And if you remember the Sylvian fissure or lateral sulcus, a lot of our language-related processing occurs around that neuroanatomical landmark. They call it parasylvian cortex, which just means cortex that's around the Sylvian fissure. So some of the notable cortical areas for language include Wernicke areas, or Broadman area 22, and Broadman areas 37, 39, and 40, which is associated with processing phoneme sequences and words. Phonemes being the smallest parts of language that carry meaning. Typically that's individual letter sounds. So damage to these areas results in what's called fluent aphasia. You'll have disturbances in speech comprehension with well-articulated and meaningless speech. So they call it word salad. I think there was an episode of House where they displayed this, where House was asking one of his patients a question, and he said something like, "I couldn't tackle the bear," which was not meaningful for that situation. This type of injury also tends to disrupt repetition and naming abilities, and they display what's called paraphasic errors, which are language output errors. That can include using the wrong word in the wrong situation, or adding in syllables to a word that aren't there, or mixing up syllables within a word. These brain areas are also associated with writing and lexical semantics, which is the ability to identify meaning from phoneme sequences via connections with supramarginal and angular gyri. And connections between the visual parts of the inferior temporal lobe are associated with grapheme, which are the written displays of speech that are then converted into phonemes mentally so that we can recognize words while reading. Other notable cortical areas for language include Broca's area, or Broadman areas 44 and 45, and the motor cortices. And those are associated with the articulation of speech sounds and the production of words and sentences. Damage to these areas results in non-fluent aphasia, which is trouble with articulation and speech production with intact comprehension abilities. The speech for these individuals is often halting and missing in functional words, things like articles. That's the, is. So they tend to be able to produce more caveman-like speech. So the processing of syntax and grammar is associated with reciprocal connections between these areas and some of the prefrontal, premotor, and supplementary motor areas. Another important anatomical feature associated with language is the arcuate fasciculus, which is a white matter tract that connects Wernicke's area directly to Broadman's area. Damage to this structure results in conduction aphasia, where someone shows deficits in their ability to repeat phrases despite otherwise intact speech production and comprehension. So these injuries are all associated with damage in the language dominant hemisphere. When damage occurs in the non-dominant hemisphere, it can result in a prosodias. So if you suffer damage to the inferior right frontal lobe, that can disrupt your ability to emote while you're speaking. or to have your speech carry emotionally related expression. If the damage occurs in the posterior temporal parietal area of the non-dominant hemisphere, that can disrupt your ability to comprehend the prosody of other people's speech. So that includes their inflections or the emotional content of how they're phrasing things. There's a few other notable aphasias, including anomic aphasia, which is impaired access or storage to what's called a person's lexicon. So a lexicon is your general knowledge and information about words and how they're used. And this can reduce single word production for common nouns, but repetition and comprehension tend to be spared when this occurs. And this is caused by injury to the inferior parietal lobe or parasylvian connections of the language dominant hemisphere. There's also transcortical motor aphasia, which results in someone having trouble with speech initiation and maintenance. And it often looks similar to Broca's aphasia, with comprehension and repetition being spared. And this is due to damage in deep white matter tracts that connect Broca's area to the parietal lobe. So there's no direct injury to any of the parasylvian language structures. So transcortical motor aphasia tends to be associated with anterior watershed infarcts. Infarct is just a type of stroke, and watershed just means that it's on the edges between the structures that are supplied by either the anterior cerebral artery, the middle cerebral artery, and the posterior cerebral artery, related to vascular blood supply to keep the brain nourished. There's also a transcortical sensory aphasia, and this displays as trouble with word comprehension, similar to Wernicke's aphasia. However, repetition is spared in these individuals, and they display what's called echolalia, or unsolicited repetition of phrases or sounds made by others. And this is due to damage to white matter tracts connecting the parietal and temporal lobes, and it's associated with posterior watershed infarcts. Additionally, language deficits can be caused by damage to parts of the corpus callosum, as well as the visual cortex, and that can result in things like alexia without agraphia, which is the inability to read with spared ability to write, optic aphasia, which is the inability to name objects visually, but you can still use the objects effectively, inability to name colors, or called color agnosia. Another language disruption is called pure word deafness, which is the inability to understand language with intact identification of nonverbal sounds. So you can recognize birds, bells, or other sounds, but if someone's speaking, it doesn't make sense. And this is caused by a disconnection of Wernicke's area from the left and right auditory receptive areas. Now we're going to be moving on to neuroanatomy related to frontal and executive abilities. So the frontal lobes have extensive connections to other cortical as well as subcortical areas. So this includes extensive connections with the parietal lobes, which contribute to things like attention, proprioception, or your knowledge about where your body is in space, and visual motor integration. Additionally, there's connections with the temporal lobe, which contribute to things like memory, as well as emotional processing. There's also important connections with the thalamus, as well as the basal ganglia, which helps us control our attentional processes as well as our movement. These connections allow for modulation as well as volitional control of a lot of these other cognitive systems, and it enables us to complete goal-directed behaviors. So important systems on which our frontal lobes can modify our cognitive skills include things like our perceptual system, our emotional systems, as well as our action-oriented systems. And this makes sense because you can direct your attentional system to specific aspects of stimuli if you so choose, as well as you can influence the emotions that you experience using your higher order cognitive abilities. And you can modify and modulate your behavior or actions based on planning and the situation that you're in. The frontal lobe connections are often in what are called loops, and some of the prominent loops include the cortical, striatal, palatal, thalamocortical loops. So it's loops of connections within the brain that provide processing and feedback information back to the cortex itself. These loops are heavily involved in behavioral activation as well as selection of what behaviors you're going to choose, as well as what aspects of your environment you're going to engage with. And there are functionally different loop pathways for the different cognitive and emotional domains. And each loop differs in the frontal regions that are involved, as well as some of the subcortical regions in the basal ganglia and the thalamus that are involved. And the basal ganglia plays a critical role in modulating both thalamic as well as cortical activity within these loops. And if you look at the graphic on the right, it looks kind of complicated, but it highlights both the loops and what kinds of signals the connections are transmitting, whether they be excitatory or increasing the likelihood of the next nuclei to fire, or inhibitory or preventing the next part of the loop to fire. So if you look, it starts in the top left from the cortex, and that goes to the striatum in the basal ganglia. And the striatum has inhibitory connections to a variety of areas, including both parts of the globus pallidus. And from the globus pallidus, these connections may move to other nuclei or may move up into the thalamus. And then the thalamus connections feed back toward different parts of the cortex. This chart highlights some of these cortical loops for some of the different cognitive systems that exist within the brain, whether that be response, reward, and learning, emotional regulation, the higher order executive functions, eye movement that are related to cognition, or just general motor control. So we're not going to go through all of these, but I think it's worth going through one. So we'll start with reward and learning. And if you look, the frontal areas that are involved are the lateral orbitofrontal cortex, the temporal cortex, as well as the anterior cingulate. And then these cortical areas project to the ventral medial caudate head within the basal ganglia. And that ventral medial caudate head transmits to the internal globus pallidus and the substantia nigra pars reticulata. And those nuclei output to the medial dorsal and ventral anterior thalamic nuclei, which in turn feeds back to that orbital frontal cortex. And if you look at the other columns, you can see those types of loops occurring. We're going to talk about some of the specific cognitive domains that frontal lobes can exert control over. So we're going to start with attention. And according to Posner and Rothbart, there's 3 interconnecting attentional systems. So there's orienting to stimuli. So that would be being able to locate where a certain stimuli is in space. For example, if you're in a crowded room with a lot of noise and someone calls your name, you would be able to know through this system whether that name is being called to your right, to your left, or behind you. And this helps tune our perceptual systems to relevant information from sensory input. And this involves what's called cholinergic connections, or connections utilizing acetylcholine as the primary neurotransmitter between the superior colliculus, which is part of the mesencephalon, and the pulvinar thalamic nucleus, as well as the posterior temporoparietal cortex and frontal eye fields. The next attentional process or system is alerting. So this is our sensitivity to incoming stimuli. It would help you pick out hearing your name in that crowd versus hearing the name of someone else, because you would be more attuned and alert towards having your name called. And this system involves what's called noradrenergic connections, or neuronal connections that predominantly utilize norepinephrine as a neurotransmitter along the ascending sensory inputs for the thalamus. That said, target detection involves a lot more of the ventral frontal parietal systems. And finally, the third system is executive control, and this involves monitoring and resolving conflicts between our thoughts, feelings, and behaviors. So it essentially allows us to sort through the information that's being sent back to the cortex and act on it in a meaningful and useful way. And this process involves dopaminergic connections, or neurons within the brain that predominantly use dopamine, including brain areas like the anterior cingulate cortex, as well as the dorsolateral prefrontal cortex. And you can read more about these different executive attentional systems within this paper by Posner and Rothbart in 2007. So there's extensive interaction between the frontal lobes and some of the more posterior perceptual systems, and these frontal lobes can exert control over how the information is processed in those posterior perceptual systems. When the frontal lobes exert this influence, that's called top-down processing. And this particularly occurs along the dorsal frontal parietal system. So if you can remember the dorsal and ventral streams within the brain as far as how information is transmitted through both the posterior and frontal areas, the dorsal is more superior or along the top of the brain. There's also what's called bottom-up processing, which involves sensory information that's pre-processed by some of the lower brain areas like the superior colliculus and pulvinar, and that tends to be biased towards what's called salient stimuli. So sometimes there's stimuli that evoke such a strong reaction that it can pull our attentional system toward it. And complex attention throughout our day-to-day involves both some of these top-down processes where we're directing our attentional system to focus on this but not that, and some of these bottom-up influences where there's powerful stimuli in our environment that draw our attention so that we focus on it more. The frontal lobes also play a role in working memory. Goldman-Rackick used a lot of primate studies, and he demonstrated a working memory subsystem that's related to the dorsal lateral prefrontal cortex. And he found that these neurons selectively activated during delay periods of working memory tasks, which he thought indicated was to help keep the information during these tasks alive or online. And he found that Broadman area 46, or the arcuate sulcus in the dorsal prefrontal cortex, codes specifically for spatial information, and found that the inferior frontal convexity, or Broadman area 12, that's found in the ventral prefrontal cortex, responds selectively to object-based working memory tasks. So this reinforces the idea that there's two streams within the frontal lobes for processing of information, whether it's related to spatial information versus more object-oriented information. So there has been some debate about how to conceptualize the dorsal and ventral processing differences. So as I had mentioned, one conceptualization is this domain specificity, where the dorsal aspects process that spatial information and the ventral aspects process more information about the object. There's also another conceptualization, which is process specificity, where the dorsal aspects are involved in things like mental manipulation and reorganization, whereas the ventral aspects are involved in sequential organization and storage. One way to conceptualize these differences would be the dorsal would be more related to if you had a list of letters and numbers and you had to put them in order from smallest number to biggest number, or in alphabetical order as far as letters go. So the dorsal aspects would be more involved in that kind of sequencing process. Whereas if you had to regurgitate a phone number, that would involve more of that ventral aspect, where you have to learn and recall the phone number as you learned it in the same order and sequence that you learned it. So there's evidence to support both conceptualizations. So this chart, once again, highlights these dorsal and ventral pathways in both our posterior visual systems as well as along our anterior frontal areas, with the images on the left being the ones that we've been recently talking about, showing the movement of information along both the dorsal and ventral parts of the frontal lobes, whereas the right has more to do with the posterior progression of information from our primary visual cortex. The next topic we're going to talk about is neurochemical projection systems. Briefly introduced that there's a variety of neurotransmitters that are active within the brain that are released into the synaptic gap, and that those can play an important role in either causing excitation in the postsynaptic neuron or inhibition of the postsynaptic neuron. So these neurotransmitters play an important role in various cognitive processes. So we'll go through them one by one, but we're not going to talk about all the different neurotransmitters that exist. So the first one we're going to talk about is acetylcholine, and that's been implicated in attention, learning, and memory. And it's the primary neurotransmitter of the peripheral nervous system, so it plays a limited modulatory role within the central nervous system. And these connections are often called cholinergic connections, to indicate that these connections often produce and preferentially release acetylcholine in their synapses. If you look at the chart down below, you can see how some of these connections run within the central nervous system. So for example, one connection starts in the pontomesencephalic areas, and it projects and connects to the intralaminar nuclei of the thalamus. And there, it causes excitation of the thalamocortical projections. And that's how it modulates different cognitive processes. This pathway plays an important role in attention, memory, and overall regulation of these thalamic outputs. And if you look at this graphic, you can see the cholinergic pathways with some notable nuclei and the extent to which these connections spread throughout the brain. Additionally, related to acetylcholine, there's two postsynaptic receptors that bind with acetylcholine. And we come up with their names based on other chemicals that preferentially bind to those receptors. So one of these receptors is called nicotinic because they found that nicotine preferentially binds to this specific acetylcholine receptor. Nicotine has a similar shape to acetylcholine, and it facilitates the rapid transmission with sympathetic and parasympathetic nervous systems. So we haven't talked much about sympathetic and parasympathetic nervous systems, but essentially they're the aspects of our nervous system that prepare us for stress or to manage situations versus the ones that calm our bodies down so that we can relax and digest food. The other subclass is called muscarinic. It's based on muscarin, which is a compound that's often found in some deadly mushrooms, and it helps mediate some of the main cognitive effects found within these cholinergic pathways. There's a few drugs that have anticholinergic properties, which means they prevent the activity of acetylcholine within the brain, and these include antihistamines, first-generation antipsychotics, as well as tricyclic antidepressants, which if you're disrupting these cholinergic pathways, that can impact the aspects of cognition that are associated with acetylcholine. So things like attention, memory, learning, those kinds of things. And they've found that elderly or those with a lower cognitive or cerebral reserve are much more susceptible to these anticholinergic effects when they take these medications. The next one we're going to talk about is norepinephrine or noradrenaline. And this system is involved in attention shifting, general arousal, mood, sleep and wake cycles, and possibly pain. And the connections involved in this system can be both inhibitory or excitatory. So they can increase the likelihood of the postsynaptic neuron from firing or decrease the likelihood of the postsynaptic neuron from firing. Norepinephrine has a well-established role in various neuropsychiatric disorders, including things like depression, bipolar, and obsessive-compulsive disorder. And the systems that use acetylcholine or serotonin can interfere or inhibit the activity along this norepinephrine system or noradrenergic system. They found that ADHD drugs often increase norepinephrine as well as dopamine levels within the brain. And some of the medications that are implicated in this include methylphenidate, which is the generic name, which includes trade names such as Ritalin or Concerta, as well as amphetamine dextroamphetamine, which trade name is Adderall. Just a note that amoxetine, or trade name Strattera, only affects this noradrenergic system, as it's a specific norepinephrine reuptake inhibitor, meaning that it prevents the removal of norepinephrine from this synaptic gap, which allows the norepinephrine to continue to stimulate the postsynaptic neuron. There's a couple of nuclei associated with norepinephrine production, and they project throughout the cortex, and once again are involved in things like attentional shifting, arousal, mood, and moderating our sleep-wake cycle. And this graphic shows where these nuclei are located and the extent to which they project throughout the brain. The next neurochemical projection system we're going to talk about is serotonin, also called 5-hydroxytryptamine, or shortened to 5-HT. And serotonin is involved in mood, both general as well as sexual arousal, pain, respiration, managing temperature, as well as motor control. And it's been found to play a role in various psychiatric syndromes, including things like depression, anxiety, obsessive-compulsive disorder, aggressive behaviors, as well as eating disorders. It has 4 receptor subclasses, but we're not going to get into those. And the majority of antidepressant and anti-anxiety drugs actually alter the availability of serotonin within the brain. So some of the common selective serotonin reuptake inhibitors, which remember a reuptake inhibitor prevents serotonin from being removed from those synaptic gaps, which allows it to continue to operate longer than it otherwise would have, include fluoxetine, trade name Prozac, and sertraline, which is trade name Zoloft. Venlafaxine, trade name Effexor, affects both the serotonergic as well as the noradrenergic systems. And if you look at the chart below, you can see where the primary nuclei associated with serotonin production are located and see their projections as well as how those projections influence different cognitive abilities. There are also serotonin receptor 2A antagonists that are common medications, including things like trazodone or Deseril, as well as mirtazapine or Remeron. These medications prevent the activity from occurring in the specific 2A serotonin receptor. If you look at this chart here, you can see where some of these nuclei are located and the extent to which this serotonergic system projects throughout all of the brain. The next neurochemical projection system we're going to talk about is dopamine, and it's been implicated in things like motor functioning, memory, our reward system, our executive abilities, attention, sexual behaviors, as well as lactation and menstruation. And there's three primary subsystems for this dopaminergic system. One is called the mesostriatal system, and damage to that system is associated with various motor difficulties like Parkinson's disease. The next is the mesolimbic system, which is associated with our reward processing and is found to be disrupted in individuals that display various addictions. Additionally, overactivity within this mesolimbic system is associated with what's called the positive symptoms of schizophrenia. So that's things like hallucinations or delusions, where people are having the extrasensory experiences or delusional beliefs about things occurring in the world. The final subsystem is the mesocortical subsystem, which is thought to contribute to the negative symptoms of schizophrenia. So a negative symptom would include things like social withdrawal or being less active in caring for yourself, as well as dysexecutive syndrome, as well as bradykinesia. Bradykinesia being just generally slowed movement overall. And if you look at the chart down below, you can see some of the specific nuclei that are associated with these different subsystems, as well as where those projections go and some of the specific cognitive areas that those subsystems are implicated in. So if you look at this graphic, you can see where the dopaminergic system exists and some of its projections. So the positive symptoms of schizophrenia are typically treated with drugs that are antagonistic to the dopamine serotonin receptor 2A. So they prevent that receptor from being active. And these include clozapine, or trade name Clozaril, quetiapine, or trade name Seroquel, and risperidone, or trade name Risperdal. So currently there's 63 other molecules that are confirmed to or thought to act as neurotransmitters within the brain. So I'm not going to go through all of those. Some of the other most common ones that we haven't talked about include gamma aminobutyric acid, or GABA, and that's involved in memory, anxiety, and arousal, as well as neuromodulation. And it's the primary inhibitory neurotransmitter within the brain, and it has broad connections throughout the brain. So many anti-anxiety drugs enhance this GABAergic neurotransmission. The other major neurotransmitter is glutamate, which is involved in learning, memory, long-term potentiation, synaptic and neuroplasticity, as well as neurogenesis. And glutamate is the primary excitatory neurotransmitter. So within the glutamatergic system, it makes the postsynaptic neuron more likely to have an action potential. If there's a dysfunction in this glutamatergic system, it can lead to what's called excitotoxicity and neuronal death. So excitotoxicity is essentially when a neuron is so active that it dies from being overly active. And that process has been found to occur in things like stroke or other kinds of neurodegenerative disorders, like Alzheimer's disease or amyotrophic lateral sclerosis, also called ALS. So there's a particular receptor for glutamate that plays an important role in what's called long-term potentiation and neuroplasticity. It's essentially the way that our neurons reorganize themselves to either be more sensitive to particular types of neurotransmitters or be less sensitive to them, and this can essentially allow us to rewire our cognitive functioning over time. So this receptor is called the N-methyl-D-aspartate, or NMDA glutamate receptor. So there's a specific medication called memantine, or trade name Namenda, that antagonizes this NMDA receptor, and it's commonly prescribed for Alzheimer's disease. And it's thought to be effective by inhibiting some of the pathological aspects of glutamatergic activation and helps preserve general physiological activation of NMDA receptors. There are some common side effects related to these various psychoactive drugs, as well as other medications that aren't used for the explicit purpose of psychoactive intervention. So for example, antihistamines often produce a sedatory effect as well as disrupt cognitive efficiency, because antihistamines disrupt our cholinergic and serotonergic systems. Benzodiazepines is another class of drugs that has these side effects, and it disrupts our memory as well as psychomotor speed by causing certain GABA receptors to be overly active. As well, various drugs used to treat things like hypertension, cardiac arrhythmia, glaucoma, or migraine headaches can have broad effects on the beta adrenergic systems, and that can further influence our cognitive functioning. If you'd like to know more about any of these topics, these are some great books to look into. So the first book is the Clinical Neuropsychology Study Guide and Board Review by Stuckey et al. And the second is Neuroanatomy Through Clinical Cases by Hal Blumenfeld. That book in particular contains a lot of the case knowledge when it comes to how injuries to specific parts of the brain affect our cognitive abilities and behavior. And other books, if you want to go into much more detail and depth about neuronal functioning, neuroanatomy, as well as the biochemical functioning of neurons in the nervous system. Neuroscience by Pervez et al. Has as much depth of information as you can look for, as well if you're interested in the neurochemical projection systems and some of the pharmacological interventions involved in them. Stahl's Essential Psychopharmacology is another great book related to that topic. So that brings us to the end of this presentation today. If you're interested in more study resources, be sure to go to brainaacn.org, where you can find written and oral exam study groups, opportunities to work with mentors, mock exams as well as sample questions for the board exam written test, flashcards, as well as other book recommendations. This has been TLDR Neuropsychology. Be sure to like, comment, and subscribe. Thanks for your time, and have a great rest of your day.